Hi everyone, I'm the Plant Propagator and welcome to my channel. Today I am happy to share with you my second PowerPoint on plant biotechnology. So the topic today is going to be DNA, just a basic introduction to DNA. And I'm going to try to keep things pretty simple for you, so I'm not going to delve into too much detail unless you want to hear more detail on it, but I'm going to try to keep things pretty simple. I also did want to say thank you to those of you that have responded and provided some comments and constructive criticism. So the slides in my presentation today are all going to be numbered, so if there's specific comments or questions that you have for any of the numbered slides, you can refer to them like that. So thank you for suggesting that. I also wanted to wish everyone Happy New Year. So it's late December of 2022, and very soon it will be 2023. So Happy New Year to everyone. The other thing is that I will be returning to Southwest Florida pretty soon. So yay! Even though it's pretty warm here today in the Midwest, we had all the snow is gone. So yay again for that. And the temperatures are pretty warm, but it's still cool. It's not quite warm enough for short sleeve shirts and short pants. But anyway, I'm looking forward to getting back to Southwest Florida and seeing how my orchids dealt with the cooler temperatures. Okay, without putting things off for very much longer, let's get into the presentation on DNA basics. Okay, and we're back and ready to go. What I want to do is today, in the second lecture on plant biotechnology, is to give you an overview of DNA basics. And I guess the phrase here, DNA is DNA is DNA, what this means is that all DNAs are the same. So you can take a DNA from a frog and from a human and from a plant. And when you look at the basic components of those DNAs from different organisms, the components are the same. The individual molecules in there are the same or pretty similar. So you can actually take a gene out of one organism and introduce it into another organism. You can introduce the DNA and get it to replicate. Sometimes it responds properly. In many cases, it doesn't. But the manipulation and the study of DNA is very similar regardless of the organism. What I want to do today is then go over what is a gene, and essentially what are the components of the gene, what are the individual molecules that make up a gene. Then I'm going to give you some examples of genes so you can see that similar genes from even very similar organisms are a little bit different. So we're going to talk about that a little bit and go into that a little bit as well, not too much. Again, what I want to do today is keep things fairly straightforward and simple. I'm going to be then talking about DNA cloning and synthesis. So this has really allowed the biotechnology area to tremendously expand and accelerate, because the DNA cloning methodologies that are out there in DNA synthesis has really become pretty straightforward and pretty simple. What I want to end up then talking about today is the parts of the gene and how those different gene components contribute to gene expression. And I've worked in this area for a while. So I think I have a good understanding. And what I tend to do is focus on the areas that I know better. So we'll tend to do a little bit of that. So the first slide that I have, and I should also point out to you that on the bottom right of your presentation, hopefully you can see the numbers, and these are the new numbers that one of you asked to be included, and I think they're pretty useful. I hope you can see it, at least. It depends on how you're viewing this. The next slide that I have for you shows something that when I look at DNA and when I study DNA, I have this movie or this slide in my head. And it's not a real good representation of DNA, but it shows the one thing I wanted you to take home from this is that the DNA, as shown in this, I think this is a GIF, is always moving. So this DNA, it's kind of jumbling around, it's moving. It shows the structure DNA. It's not really accurate because it's a small, this DNA is kind of displayed as a very small circular piece of DNA. And it doesn't really look like this, and you can't see it. But what I want you to understand, what I want you to recognize is that DNA is always moving, and the components are always moving, and the proteins that interact with the DNA are always moving around, and it's a pretty rapid response and a pretty rapid, I guess, jiggling, if you want to call it that. And so you have to view this as going on, really in all organisms that contain DNA and all cells and everything like this. This is overall a really rapid process, and everything is moving around fairly quickly. And I like to show this because it reminds me of that, and I want you to be aware of that as well. Okay, so first of all, the first question is, what is a gene? And the central dogma here at the center of the slide is that DNA gives rise to RNA, and RNA gives rise to protein. DNA, I shouldn't have to tell you this, stands for deoxyribonucleic acid. It's not important. It's better that you just remember DNA. RNA stands for ribonucleic acid. Again, that's not as important as remembering RNA. I put terms in here, but you don't need to remember these terms. So the process of when DNA generates RNA is called transcription. It's the first process, and then the process where RNA gives rise to protein is called translation. I used to remember this by saying, well, TRANS is the same in both of these words, and then this is a C and this is an L, and the C is earlier in the alphabet. So transcription is first, DNA to RNA, translation is second, RNA to protein. Okay, so as you produce protein then, what happens is that proteins interact with other molecules and drive the cell machinery. So this is the central dogma. DNA gives rise to RNA, RNA gives rise to protein, and then proteins interact with other molecules and drive the cell machinery. So that really is the key to what is a gene and what genes do. And these other definitions and terms, I'm going to present them to you, but you don't really need to know them. And on the next slide, I've got, the four basic bases or components of DNA are A for adenine, T for thymine, G for guanine, C for cytosine. And they bind in very specific ways. You don't really need to know what these things stand for or how they bind, but you do need to know that when you look at this as a piece of DNA, and this also is just one strand of the DNA. It's double-stranded, but this is one strand of the DNA, and it goes in a certain direction, and there is directionality to DNA, but the order of the ATGC defines what that gene is. But really this is the order, this is CAAGGTAT. If you can't see it, fine. I just read the first few bases or nucleic acids of this sequence from P polyubiquitin gene. And this is actually the part of the gene that gives rise to the protein. All right, so you don't really need to know too much other than these are the four main components. Just remember the letters, and this gives rise, in the order and the organization of this gives rise to the specific product that the DNA is producing. When you take a look at a similar gene, so this is barley polyubiquitin, and you don't need to compare the sequences, but it's a little different. There's a lot of sequence homology or sequence similarity. I'm going to back up a slide between the P polyubiquitin gene and the barley polyubiquitin gene, and that similarity gives the similar gene function, but there are differences in the sequence, small differences in the sequence that identify really what organism this comes from. Okay, so taking this, let's dissect this a little more, and taking this a little further, this is a part of a soybean polyubiquitin gene. And I studied this gene for much of my career. And this GM up here stands for glycine max. UBI is the polyubiquitin gene. So this is GAMUBI, and we've studied this gene in my laboratory a lot. And some of these, there are capital A's and small a's, and they're then regions of this DNA that are highlighted. And these regions of this DNA have defined functionality. So really the best way to study this is to take these DNAs apart, put them back together, and mix and match and study these things. There are now programs where you can take these sequences of ATGC, throw them into a program, and it'll tell you what the gene is and what the various components probably are. It's not 100% guaranteed, but it kind of gives you an idea of what these different pieces and parts are based on the information that's already out here. So this actually happens to be part of a soybean polyubiquitin gene, and I studied this, and I know what most of these pieces and parts are. Not all of them, but I know what these pieces and parts are. And so you can study these things if you again take them apart, put them back together, and see what they do. And like I said, again, these have been tagged and annotated for having various functionalities that the computer will do, but it has to be really done it has to be analyzed appropriately through some of the transgenic technology that we'll get to later on in this course. Okay, so what I want to I'm going to shift gears a little bit. I think get an understanding of the makeup of the DNA and how the various pieces and parts contribute to gene expression. But now I want to talk about the cloning techniques. How are DNAs cloned, and how are they analyzed, and how they are studied in the laboratory. And what I want to talk about, the first thing, is restriction enzymes. And restriction enzymes, we'll get into this in just a second, they recognize very specific sequences in the DNA. So it has a very specific target sequence that it binds to. And this is an enzyme, so what it does is it digests or cuts the DNA very precisely. I want to then explain to you a little bit about plasmids, how these DNA pieces and parts can be put in these other carriers of DNA called plasmids and then cloned in bacteria. I want to talk with you about PCR, which I think you've heard a lot about, and talk about a little bit again about DNA synthesis. Okay, so the first topic area here is restriction enzymes. And what I said is that restriction enzymes recognize very specific sequences within the DNA. So this is a very small piece of double-stranded DNA, and here it shows the binding of the A to T, the G to C, but this is the sequence right here. And the restriction enzyme that we're looking at recognizes this sequence right here, which is in reality right here on the little bit of the left side of this sequence. All right. And what I want to do is explain to you about cutting and sewing of DNA components using restriction enzymes. So restriction enzyme, again, is an enzyme. It's a protein that's going to recognize this specific sequence right here. And then what it does is it comes in here and it recognizes this specific sequence, and this is a restriction enzyme that's one of the most common ones. It's called ECO-R1. The ECO stands for the organism that it was cloned from, and here this organism happens to be Escherichia coli, E. coli, which is a common gut bacterium. And this then recognizes this specific sequence. And what happens when this enzyme comes in, when you mix this DNA with this restriction enzymes, the proper buffer, the proper everything else, is it cuts the DNA and leaves this exposed end like this. The next step in this overall process then is to remove, so this is the part of the DNA that we're trying to clone, and this is the DNA that it came from, and then the next thing is to I'm sorry, I had that backwards. This is the DNA that we're trying to clone. So what we did, I'm going to back up a slide, is we put a new sequence over here. And this sequence is the same matching sequence, AATT, which will bind to TTAA. And then we add another enzyme called ligase that will then allow these pieces of DNA. It essentially will allow you to sew them together, shown right here. All right, so this is what happens when you cut and sew DNAs together very precisely using this approach. All right, so what you can do with this then is clone DNA pieces as fragments of DNA into these circular DNA molecules called plasmids. And so this is one of the most commonly used plasmids. It's called PUC19. The UC stands for the University of California that designed this plasmid. And what you do with this is you take very specific fragments of DNA, you cut this plasmid open very precisely in this region, and then you can insert pieces of DNA in here. This whole plasmid then is introduced into a bacterium, and that plasmid will then replicate. And what it allows you to do is generate large numbers of very precise copies of this plasmid containing the introduced DNA. So DNAs are cloned and manipulated in plasmids, which are then grown in bacteria. The bacteria also have their own chromosomal DNA. So these are small circular DNA molecules that are outside of the chromosomal DNA that are grown in these bacterium. But you can generate a lot of this plasmid DNA that can then be manipulated. These are not as important as they used to be. They're still useful. They're still valuable for certain things, but you can now, there are other ways to generate large amounts of DNA, of cloned DNA, and we'll talk about that in a second. Plasmids have to have the ability to replicate, so they need an origin of replication. The polylinker is where the introduced DNA goes, and there's other components of the plasmid that make it useful or that are necessary so that you can grow DNA, large amounts of DNA in the circular plasmid that is then grown in this bacterial cell. All right, so that's essentially, I just wanted to give you a basic overview of how DNA can be very precisely cut, sewn back together, and then propagated or cloned in these plasmids, which are then replicated in these bacterial cells at very high efficiency. Okay, so moving on a little bit, what I want to shift gears a little bit and talk about PCR. And PCR stands for, and I don't have it written down here, polymerase chain reaction is what PCR stands for. And what PCR allows you to do is to very precisely replicate fragments of DNA. So PCR relies on using various primers, and primers are small DNA fragments that then attach to a larger piece of DNA. So you have a forward primer, a backwards primer, and these are separated and reannealed by high temperature and then followed by low temperature. High temperature causes the DNA to kind of split apart. Low temperature, so you have these strands, it's melted, these strands are separated, and the cooler temperature, these primers will then bind to these targeted pieces of DNA. And what it allows you to do is generate large numbers of these very small DNA fragments. The DNA fragments, and there is a lot more information about this that I want to share with you. There's a lot of good information aside, of course, from my presentation today that's on YouTube, and I'll give you links to those, and I'll tell you a little bit about those in a second. What the PCR does is it generates using target DNA, it generates an amplicon. So the amplicon, I'm going to back up a slide, is this piece of double strand, a very small fragment of DNA that is generated from the parental strands of DNA shown in this previous slide. And so what you can use is one copy of a target through a lot of generation of amplicon is amplified. That's the original part of the word. So you can generate 1 billion copies after 30 cycles. And you don't really need that much DNA, template DNA, in order to generate that copy. It's used, this is a great technology, a great tool. Your PCRs are you need very small volumes, 10 to 50 microliters, which is really not much. It's like 5 to 10 drops of water is what the equivalent volume is. And it's easier. What you do is this relies on heating and cooling in these machines called PCR machines. And what it does is it generates very specific results, which are these very precise amplicons. And these amplicons you can just see what you have just based on the size of the amplicons, and then you can also sequence the bases in the amplicons to get really good detail on what's there. For most of the PCR tests that are done, the size of the amplicon is really revealing. You can use partially degraded DNA as long as there is some intact DNA present. But PCR, as you've known and as you've heard about, it's really routine and very standard protocol for amplifying very small pieces of DNA. If you want to learn more about this, there's some great YouTube videos that you can go to here. I'll put the links to these in the video description of my video today. I'll also try to link these in my YouTube video itself, but I'm not sure if I'll be able to do that. I can link to them here in my PowerPoint, but I'll do more. And this is one of a frame shot of, I think, from the second video. But both of these are pretty short, and they show you how PCR is used using a video that I just can't assemble, using a video that shows you schematically how PCR works. All right, but essentially the bottom line from PCR is that if sequences are known, if a sequence is known, primers can be designed, and the primer is designed to bind to the parental DNA, and then PCR is used to generate this amplicon, which can subsequently be cloned, analyzed, modified, reintroduced, or studied. So PCR is just, again, it's a really great tool. And then these DNA components that you can generate can be dissected and analyzed. Okay, so big deal. Now what? And I want to go into a little bit more detail on what are the parts of the gene. I want to break this down. We saw previously the sequences that were highlighted in the different colors, and I said the highlighting means different things. And what I want to go to now is just a really a little bit of detail of what are the components of a standard gene and how they function. And this is in general what you get when you look at a gene right here. This is in really simple terms. Again, this is it's unidirectional. It goes from the left to the right. This is all stretched out, and normally, like I said, the DNA is folded and it's convoluted, and this is just really a very simple schematic. And the three main components of a gene are the promoter, shown here, the coding region, and the terminator. The coding region, I want to start with this. The coding region is what actually gives rise to the protein itself. So this is the DNA sequence in here that gives rise to the RNA, and the RNA then gives rise to the protein, and the protein is the enzyme or the protein that has functionality. So that's this region, the coding region. It encodes for a DNA that gives rise to RNA, which then in turn gives rise to the protein. So this is the core of the gene, is the coding region. The promoter, what the promoter does is regulate the gene. So the promoter tells the machinery in the cell If this gene needs to be turned on in certain tissue, whether it's whether it's needs to be induced under certain conditions in plants, in the light, in the roots, in the stem, in the leaves, whether it should be induced in response to high temperature, low temperature, high salt. So inducibility refers to the promoter and when this gene is turned on to give rise to the RNA and the protein. In the plant area and in the plant transgenic area, you can use homologous and heterologous region promoters. So you actually can take a promoter from corn and introduce it into orchids, and it'll likely have good functionality. This all needs to be studied, though. You can also take coding regions from this. So you can mix and match what it says on the bottom here. You can mix and match components, DNA components, to see how it works and when it works and if everything is going as it should be. All right, I'm sorry, I'm getting ahead of myself. Enhancers are fragments of DNA in the promoter region that enhance expression of the main coding region itself, or of the gene itself. Within the coding region, you can have leaders and signals that will determine where the RNA goes, and there are other signals that are in here. There are introns and exons. Introns is it's labeled and named intron because it's intervening. It's actually processed out, but here's some components. It gives rise to the RNA, but then it has an effect on RNA stability. And then the exon, however, is those are the EX stands for the expressed part of the gene. The terminator, as you might imagine, is not the former governor of California, but the terminator is the end of the gene. And it also contributes to expression of the gene and then stability of the message. And the message refers to the RNA. So the messenger RNA is what the DNA gives rise to, and the terminator can affect message stability. So what we do in the laboratory then is mix and match DNA components and introduce those and study those and see what effect those components will have on gene expression, and even see what the gene itself does. And you can mix and match and do a number of different things. And this is really one of the exciting outcomes of plant biotechnology. Okay, so each gene is regulated by its own promoter, and the promoter then determines the timing, expression, and specificity of gene expression. And these are what's shown here in this cartoon is components that bind to the promoter region that lead to transcription of this gene. And so you have these components that are called, you don't need to remember their transcription factors, but there's a number of different components that will bind to the promoter region that will turn on gene expression. And that's what you can remember that's what you should remember from this slide. So the promoter, so through the gene and gene manipulation, gene expression can be increased, decreased, modified in certain ways. So you can insert whole new genes using the transformation or transgenic approaches. And we'll have much more detail on that later. But then the exciting thing is through genome editing, you can take existing genes and modify or mutate the existing genes to modulate native gene expression, and that's exciting. So you can make mutations in the coding region, that's the gene itself. You can that's all the gene itself. You can modify the promoter, which then allows you to increase or lower the expression of the native gene, and then you can change the gene itself in small ways using genome editing approaches. So there's a number of really exciting things that you can do with gene expression and with even the specificity of the gene through both transgenic approaches and through genome editing approaches. That's all I wanted to share with you today, is to kind of give you a very simple, basic overview of genes and how they function and what the gene components are and how they're manipulated in the laboratory. Again, I can go into much more detail, but I wanted to keep things pretty simple. If you have any questions on this or any comments on this, whether it's too simple for you, whether you want more detail, or whether it's too much and slow down, again, please put those comments on YouTube, and I'm happy to comply. Next time, what I want to talk about, moving on, is the third lecture will be on tissue culture and plant cloning. And again, what I'm doing is I have these individual lectures or presentations, which I could talk for hours on each one. I'm really simplifying and cutting them down to make it hopefully more digestible and more interesting to you. So next time, again, I'm going to be talking about tissue culture and plant cloning, and just give you a basic overview of this. And I'll try to bring in some orchid emphasis, but the same technology, like DNA is DNA, what I talked about in the first slide. The same approaches are used in plant tissue culture and plant cloning. The same approaches are used regardless of the plant types. And what it does for me is has allowed me throughout my career to jump from plant to plant to plant using very similar approaches. Okay, so that's all I have for today. I hope you enjoyed my second lecture on DNA basics, and I hope you join me for the third lecture on plant tissue culture and plant cloning. Okay, that's all I have for today. Happy propagating.